Before we get into it, I just wanted to go over the format really quickly. So there will be a 15-minute Q&A session afterwards. So if you have any questions, you can, through the participants box, check yes or raise your hand. And Jason, who is also on the call, will call on you and you can unmute yourself and ask your question. If you're a little shy, you can always use the chat box and Jason can read off of that. And now I'll hand it off to Professor Tom Bayer, who is the director of the Stanford Photonics Research Center, and he will introduce our speaker for today.
Oh, thank you, Rebecca. First of all, let me thank Rebecca and Jason for helping to organize this seminar. This is the first in a series of seminars that'll be sponsored by SPRC, the Photonics Research Center, over the remainder of this year that will focus on key technologies for measuring and monitoring our rapidly changing environment. It's my pleasure to introduce Ken Johnson, who is a senior scientist at the Monterey Bay Aquarium Research Institute in Moss Landing. His research focuses on the development of novel chemical sensors, which can be deployed for extended periods in the ocean. Much of the focus in his laboratory is on implementation of Biogeochemical Argo, a global observing system using these sensors on profiling floats. I must say that the Argo system is my prime example of what we need to deploy worldwide to really understand what's going on with climate change. And I'm very pleased to have Ken here to tell us about the exciting work he's been doing. So I'll turn it over to you, Ken.
Okay, great. Thank you for the introduction, Tom, and Rebecca, and all. Thank you for the invitation. This is a topic very close to my heart and love to talk about it. So let me just get organized here and make sure that we have the right display settings. Okay, is that correct? Okay.
Yes, it is.
So I'm going to talk to you about Argo chemical sensors observing ocean health with a global network of sensors on robots. Just give you a quick introduction to where I'm coming from. So I'm at the Monterey Bay Aquarium Research Institute, founded by David Packard of Hewlett-Packard. Dave and his daughters founded the aquarium. Dave became interested in ocean technology and decided to have his own place. And although he passed away in the 90s, we carry on. The mission of MBARI is development of better instruments, systems, and methods for scientific research in the deep waters of the ocean. About 200 staff, research engineers, operations. We run several ships, a fairly extensive outreach and education program, primarily supported by the David and Lucille Packard Foundation, although much of what I'm going to talk to you today is actually supported by the National Science Foundation. So brief outline, this bit of introduction, the need for an ocean sensor network, a little bit about profiling floats, the robots we use to carry our sensors, a bit about the sensors we deploy, and then a discussion about next steps in observing the ocean. Most of the examples I'm going to give to you come from a program that is ongoing. It's gone through 6 years now, in its 7th year with the Southern Ocean Carbon and Climate Observations and Modeling SOCOM program. We've deployed 200 profiling floats with sensors in the Southern Ocean. It was, when we started, the least observed ocean in the world. I now argue that it is the best observed ocean in the world. Was successfully renewed for this program for 4 years, and 120 more floats. We're in the first year of our renewal. You can read more about this, socom.princeton.edu. And that success of SOCOM has led to the recent funding of the Global Ocean Biogeochemistry Array, which to some extent is taking SOCOM now globally and deploying 500 floats over the next 5 years globally. The blue dots here are what 500 floats might look like when you tell MATLAB to put dots in the ocean randomly in water deeper than 2,000 meters. Funded in November, and you can read about go-BGC, go-BGC.org. And so the examples, most of the examples I'll show come from SOCOM, and we are heading towards a global network. As I said, funded by the National Science Foundation, Office of Polar Programs at NSF. Much of the sensor development work was funded by NOPP, the National Ocean Partnership Program, and significant amounts of money from NOAA and NASA as well. So those two programs actually operate in the within the framework of a much bigger program called Argo that Tom alluded to. Argo is a network of almost 4,000, today 3,885 operational robots in the ocean. Their mission is to monitor the heat content of the ocean to assess whether the ocean is warming or not. And I would argue we actually now have better metrics for the warming in the ocean than we do on land. There is not a terrestrial network that has near the completeness of the Argo network in the ocean. I'll show a little bit more about that. It's an international teach you in graduate school is how you work in an international program. There is a huge amount of effort. About 40% of the global ocean belongs to some other nation, and the international agreements required to instrument them are interesting. And so this is just a sampling of the nations that are involved. Goes from Australia and Bulgaria all the way up to the UK and the USA, which does about half of the 4,000-ish floats. US has deployed about 2,000 of them. These are the floats operating today. As well, we're now extending, and that's really the focus, Argo, into biology and chemistry, biogeochemical Argo. This is the biogeochemical dots down here are from the SOCOM program that I talked about, as well, other US floats sort of dominating this distribution. So why are we doing this? You know, you've seen the news probably. This was the Washington Post last week. 2020 rivals the hottest year on record, pushing Earth closer to critical climate threshold. Well, one of the things that people don't realize so much is that global warming is really ocean warming. 93% of the extra heat trapped in the Earth due to carbon dioxide increasing in the atmosphere has ended up in the ocean. So if you look at where heat has been deposited in the Earth system in zetajoules, 10 to the 21 joules, the upper ocean, light blue, the deep ocean, darker blue, ice in white, soil in brown, and the atmosphere that we think of as global warming is this thinnest little layer down here. Only about 1% of the excess heat in the Earth system is actually what we experience in the atmosphere, and 93% of it's in the ocean, and that has profound impacts on the ocean. So here's the record of ocean heat content anomaly, again in zetajoules, 10 to the 21 joules. And the interesting thing are the error bars. You see the green bar right here, 95% error was large, and you come over to here and the error bars get smaller. It's the Argo array that basically has enabled us to have huge confidence in the actual warming of the ocean. So you see that little green, you can barely see the error, 95% error bar on the ocean heat content. And associated with this, so we have fantastic observational capacity now for ocean heat from the Argo array. There are associated, I could give a whole hour on possible changes in the ocean. This is one I show. Global phytoplankton decline over the past century was in Nature about 10 years ago. I've highlighted a couple of sentences. We conclude that global phytoplankton concentration has declined over the past century. Estimate a rate of decline of about 1% of the global median per year. Well, 0.99 to the 100 is 0.38. Has it, and actually this is a mistake. It's not a 38% decrease. It is 38. The ocean now, if this paper is correct, the ocean has 38% of the phytoplankton that it would have had 100 years ago. And I picked this paper. The authors are good friends of mine, but the point is extremely controversial. and really the reason I showed is because we don't, just to make the point, we don't really have that great of data on to understand what's happening in the ocean associated with the warming that's going on to the chemistry and the biology of the ocean. We need a new observing system to really be able to understand, is this real or not? And a great quote from Thomas Stocker. Stocker was the IPCC leader in the fourth assessment report, I believe it was. Given the prominent role of the ocean in the Earth system as a vital service provider, one wonders why so little attention is still paid to its physical state and the health of its ecosystems in the policy arena. Should we not step up efforts to better measure, understand, and project ocean processes? And so, basically, this quote kind of encapsulates the motivation for my research and the work that I'm going to talk to you today. We're convinced that we don't observe ocean chemistry, biology well enough. It is a vital service provider. It absorbs heat, it absorbs carbon dioxide, it provides protein for billions of people, and it is woefully under observed. And just a kind of a depressing example of that, the Noaa World Ocean Database, this is where everybody writes their data when they make observations in the ocean. They supply the data to Noaa. And what this is the annual number of profiles from ships added to the World Ocean Database. These timescales start back around 1900, run up to 2013. This was the 2013 version of it. And you see that if ships are observing platform, the salad years of oceanography were sort of in the 1980s. And since then, for heat, for oxygen, nitrate, we'll talk about, pH we'll talk about, the number of observations have just declined precipitously for a variety of reasons. But just as we're becoming aware that we really are seriously under observing the ocean, we're under observing it even more than we used to. And a lot of that has to do with the fact that historically, to observe the ocean, you had to get on a ship and go out and collect a sample and measure on the ship. And if the ship wasn't there and the scientists weren't on the ship, nobody was making observations. And these are expensive beasts to run. $30,000 to $40,000 a day is probably an underestimate today. It's probably more like $40,000 to $50,000 a day to run a major research vessel. We were talking earlier, they still only go 10 knots, right? So imagine driving across the US at 10 miles an hour. So we are deploying our robots right now off of this particular ship, the Roger Revell. It's down near New Zealand. The folks will be on that ship for close to 80 days. To get down there, they couldn't fly to New Zealand. They had to leave from the US. And, well, just sort of imagine driving to work from the Bay Area to New York at 10 miles an hour, right? Not so good. So the solution to this is robotic platforms. And I'm going to talk about the Argo profiling float. So here it is. Here's a profiling float. They're not huge, about 30-liter volume. These floats carry enough batteries. They have a buoyancy engine that allows them to descend to 2 kilometers depth. So a cycle, a profiling float like this, in its lifetime, it will spend about 20 minutes on the sea surface to phone home through the Iridium satellite network. It will alter its buoyancy and sink to 1,000 meters, drift for 10 days in the mission that we operate floats. After 10 days, it will then alter its buoyancy again, and sink to 2 kilometers, change its buoyancy, and come to the surface, making measurements. And so it collects temperature and salinity, pressure, and in our case, oxygen, pH, nitrate, chlorophyll, and a bunch of other parameters. Phones home, repeats the cycle. This float has enough power in it to make about 280 of these cycles, which works out to be a 7-year lifetime in the water. And we don't recover it, and they're not freely. They park at 1,000 meters where the currents are weaker, so they don't drift as much as if they were at the surface, and they tend not to biofoul because at 1,000 meters there's much less biological activity. And so the Argo network has totally revolutionized oceanography. I plots of the declining number of observations made from ships. So in the Argo era, which started in 1999 to today, 2000, well, this is an older plot, but to today, these are the number of profiles of temperature to at least a depth of 1 kilometer made by ships. The color scale is not temperature, but it's the number of profiles per 1 degree by 1 degree square in the ocean. Large regions of the ocean aren't sampled. Large regions of the ocean are blue, where in this 17-year period, you know, there may have only been 1 or 2 sets of samples collected. Hard to measure change if you only go once or twice in a decade, and especially because there are seasons and so on. The Argo floats now, that 4,000 float network I showed you, has turned the ocean red, basically. These are now the number of profiles, and it's typically many more than 20 profiles per 1 degree square. We're observing the ocean much more rigorously for temperature and salinity than we used to. And I just made the point, lots of international agreements, all of this data, all the data I'm going to show you by international agreement is available without restriction in real time being 24 hours at the Argo Global Data Assembly Center. That's the agreement to be able to deploy in other nations' waters. We have to share the data. No one gets it before anyone else. So we have this great system, these Argo floats, thousands of them in the ocean. The trick for us is to have chemical and biological sensors that I can put on one of these floats and let it go, and it will generate useful data for the 6-year or so lifetime of the float. And we believe the numbers. Now, a little bit about the sensors that we put on floats. Start with oxygen. A number of these sensors have been developed in my laboratory. Oxygen is not one of them. Oxygen is a commercial product. Luminescence, many of you may have heard of a Clark oxygen electrodes. That's not what these are. These are luminescence-based sensors. You have a little foil right here. You excite it with an LED behind it. If oxygen has diffused into the foil, oxygen quenches the light, and in the presence of oxygen, in the absence of oxygen, this lumiphore will emit light for a longer period than if it's quenched by oxygen. So if oxygen is 0, you get a long lifetime. If oxygen is high, you get a short time. But it's hard to measure absolute intensity of these devices. There, as if the foil loses the lumiphore, if it diffuses out, if it's photo bleached, the intensity will change just due to aging of the device. So I have to give credit to the real, the gurus that invented this. Otto Wolfbeiss in Germany, Ingo Clement in Austria were the first people. Otto's lab really, I think, invented these devices. Ingo was a student of Otto's and then applied these to oceanography and have revolutionized our ability to measure oxygen. We don't measure intensity, we measure phase shift. So if you pulse the lumiphore with a modulated light source, varying intensities, you end up with the excitation. If you measure the excitation intensity, you end up with a waveform like that. The emission intensity, due to this varying waveform, varying excitation, looks like this. And as the concentration changes, the phase shift between the peak of the excitation and emission changes. And this turns out to be extremely reliable, immune to aging of the sensor, relatively immune to temperature, just an incredibly stable oxygen sensor that you can deploy for years. And we know that they're stable. This is work that was done in my lab, because when the float comes to the surface, the sensor is penetrates through the sea surface and is exposed to the atmosphere. And we actually calibrate these devices to assess any drift by measuring oxygen in the atmosphere. So we use air oxygen to calibrate them. For hundreds of these sensors now, we can look at the drift rate for the sensors that have been in the ocean. This is first paper where we looked at 50-ish of these, and there's a histogram of the drift rates of the sensors. And you see that they tend to drift less than 1% of 1% of the output per year. Most of them don't drift at all, and we can correct for that drift. So one of the tricks in running a system like Biogeochemical Argo is to be able to assess the quality of the data and correct for problems, and that almost is where we spend most of our time is quality controlling and correcting data. But these sensors are great. They have 0.1% drift per year, which enables just phenomenal resolution on processes. We were talking earlier about floats in the Arctic. This is a profiling float. Now it's gone up and down for 4 years. In the Antarctic, the trace is right here. It's in the Weddell Sea. It started here, ended up over here. And this is the kind of information that comes back from these floats. So the data extends on down to 2 kilometers. I'm only showing you the upper 250 meters where most of the biology is happening. So this is the Antarctic temperature, going from minus 1.8, the freezing point of salt water, minus 1.8, and balmy summertime temperatures of plus 1.5 degrees centigrade. Gaps in the data, we talked about floats detecting ice. So when the float comes up, if it detects surface ice, it stops at about 20 meters depth. Tom was mentioning the work in Canada to detect optical sensors to assess the presence of surface ice. You don't want to hit it. It can break the float. In the Southern Ocean, it's actually, I don't want to call it trivial, but the ocean is much better mixed down there, and we just measure the temperature of the water. If it gets, the freezing point is about minus 1.80 degrees centigrade. If the temperature goes below minus 1.78 degrees, we know there's ice above, and we stop the float, turn around, go back down, keep cycling up and down. And when the ice eventually goes out, the float can reach the surface. Using Iridium can broadcast all of its data home in about an hour from profiles over the winter. You don't get a lat in the lawn because you need to get to the surface to get a GPS fix, but otherwise you get the whole profile and you just sort of dead reckon the position between the various times it arrives. So just an example record, the ocean is not a constant environment. You see here's a winter temperature where it's a little warmer. This is chlorophyll, we'll talk a bit about that, but phytoplankton, the amount of plants in the water, this little bit of warmer or more sunlight this winter, we have more plants, it's redder than other winters. And the oxygen sensor down here, you see the plants have produced more oxygen. And so we can really record the metabolic processes in the ocean from the oxygen evolution and have a great record of photosynthesis. And monitor the health. How fast are the plants growing? And over time, you can see the interannual variability here. So just like on land, we have cold winters, warm summers, cold summers. But over time, we will be able to assess changes in plankton productivity of the ocean. Nitrate. So huge interest in nitrate. It is the fundamental fertilizer of the ocean. So if you go to Hawaii, this is a float operating, a profiling float with a nitrate sensor operating just north of Hawaii for 2, 4, 6, 8 years. And the ocean at Hawaii, water is very clear. You can see far down, much farther than you can along the coast of California. That's because there's no fertilizer in the surface. So there's lots of sunlight, lots of water, no fertilizer. When the plants grow up here, they take the nitrate, the dissolved nitrate, convert it into particles. It sinks into the deep sea, and plants can't grow anymore. So growth at a place like Hawaii, the middle of the oceans, is really this tension between the processes that raise nitrate to the surface where the plants can get it, and then when the plants die or are eaten and sink into the deep water, that stops productivity. The interesting thing about this record was became a paper in Nature. You see all the fuzziness in here. These are all events that raise nitrate up into the surface layer. If you look, there's been a time series run from ships near Hawaii for 30 years. The record from the ships at Hawaii is a lot more boring. It's just kind of flat, right? You just don't see these events when you go out a few times a year and sample from a ship, whereas the robots, which are going up and down this case every 5 days and sampling, you see all these events that we just did not understand to be there. So how do we measure nitrate? These are optical. This is optical oxygen fluorescence lifetime. This is a spectrophotometric sensor. Nitrate has a moderate UV absorption band, pi to pi star transition. And it's down deep in the UV. So here's wavelength, here's absorbance of nitrate. Molar absorptivity is about 4,000, not super strong. But at the kind of concentrations that you see in the ocean, 30 micromolar nitrate makes an absorbance of about 0.3. That's a signal. It's pretty easy to detect. The problem is that it sits on top here, bromide, 840 micromolar bromide. It's part of the sea salt in the ocean on a different absorbance scale. The bromide is much stronger. About the time that nitrate is absorbing about 0.2 or so, the bromide's absorbing well over 1 per centimeter. So there's some math to do, but this is an optical nitrate sensor. I'll show you a bit more about the instrument. The math is actually fairly straightforward. The presumption, because lots of things absorb in the UV, that it would be impossible to get the nitrate signal deconvolved, but it really turns out the spectrum for nitrates, the UV spectrum of seawater is pretty simple. And so this black line is a UV spectrum observed with one of our sensors. So we measure, it's a diode array spectrophotometer. We have a UV light source and we measure every 0.8 nanometers. So they're in from 240 to 217 nanometers, which is where we operate. There are about 30 pixels in here, about 30 data points. And the observed spectrum is really composed of this strong bromide absorbance in red, nitrate, which can go all the way to 0 or up to about here in the spectrum, be close to equivalent to the bromide. And then the instrument has a linear baseline. That's due to organic matter, due to particles, due to instrumental drift as well, tends to produce a linear baseline. So if you add these 3 components together, you get the dashed line that fits the UV spectrum. The Beer-Lambert law would say the absorbance at any particular wavelength lambda is the molar absorptivity at that wavelength times the nitrate plus the molar absorptivity of bromide times the bromide concentration in seawater plus the slope and intercept of this linear baseline in wavelength. 4 unknowns, and if I measure at least 4 wavelengths, and I know the molar absorptivities, I can deconvolve all of these signals. We measure, as I say, not at just 4, but at about 30 wavelengths. The problem is that these are still pretty collinear signals if you look at it, and the deconvolution is not robust. To separate nitrate from bromide, the signals are just too similar to each other. And so we really take advantage of the fact that bromide is part of the sea salt background. If I measure the salinity of the ocean, I know the bromide concentration almost better than I can measure it. It's the residence time of bromide in the ocean is order of 1 million years. It's well mixed. And so I just know where it is. I measure salinity. I compute what the bromide is from this known salinity to bromide ratio. I know the molar absorptivity of bromide as a function of wavelength. So I subtract it from my signal. I transform this deconvolution. I just subtract off the bromide signal, and this is what I work with now. So I just separate nitrate from a linear baseline. That's pretty tractable. And I can do that easily. I picked this particular spectrum just to make the point there's often noise in it, and the more wavelengths, the more pixels you're working with, just the more robust your deconvolution gets. So in principle, you could get Y. So we really only have 3 unknowns. I only, in principle, need to measure, to get nitrate and the linear baseline, I really only need 3 wavelengths, but if you have 30, you are just going to be better off. To make this work, you have to know the molar absorptivity of bromide. It turns out it's a strong function of temperature and pressure for a variety of reasons. And so there's a lot of laboratory work to measure the molar absorptivity to define it. But that's, lab work, you can do that. And this is what, the basic instrument looks like. So drives folks in the lab wild, but I say it is just a UV spectrophotometer. There's a UV light source, commercial. So, whoops. I was also making the point earlier, our approach to this is not to invent things from, something like a TRL level 1 or 2 or 3, but it's to find commercial products that we can find old analytical approaches that the technology didn't really support and find new optoelectronics and apply them to them. So Heraeus makes this great UV light source. It's low power, instantaneous, turns on, launches light down a fiber optic through a probe. So here's a blow up. The probe has there's one lens here and a mirror. So launches light through, the light's reflected back to the fiber bundle and returned back down to a Zeiss photodiode array spectrometer. Back here, there's a microprocessor underneath that runs it all. There's your multimeter for scale. This is the end cap of a profiling float. This is the salinity temperature sensor. I'll talk about pH in a bit. That's the pH sensor on a float. But this is, so UV light source, fiber optically coupled, a probe to get the water flowing through and the fibers illuminate it and return light back to a photodiode array. And it works. A number of papers, you guys are, I'm just going to say, have never looked at deep sea research instruments and methods or limnology and oceanography methods or Journal of Atmospheric and Oceanic Technology, but there's a whole, literature out there on this kind of technology. I mean, we tend, I'm going to guess, operate in a little bit different worlds, but all kinds of interesting things going on in journals that I bet most of you don't look at. How do you apply this? So if you remember a year ago or so, there was a blob of hot water in the North Pacific Ocean, got very warm, killed 1 million seabirds, scientists say. There are peer-reviewed publications on this. Here's one of our profiling floats at Station Papa. Nitrate goes to 0. Nitrate goes to 0, the phytoplankton don't grow. They have run out of fertilizer, becomes like Hawaii. Here's temperature, the blob of hot water. This is what killed the seabirds. The hot water prevented the nitrate in the deep water from being mixed to the surface. Plants ran out of fertilizer, system died here. And so the record continues now. The interesting thing is ocean has gotten, so ocean cooled down here, it has gotten hot again up there. And there's even less nitrate. And there's gonna be another consequence of that coming up. You'll see more news articles about this, I would expect. So nitrate's really interesting in the ocean. There's, as I showed, there's not enough of it. On land, it's just the opposite. Here's an article from the New York Times about a year and a half ago. They grow the nation's food. They can't drink the water. The byline here is East Arosi, California. Here's Fresno, Bakersfield. East Arosi is right here. This map from the Bill Lane Center at Stanford is a map of nitrate in public small water systems. This is actually a category of water systems. More than 6 hookups and less than 100. Sort of trailer parks kind of size water systems. And this is the concentration of nitrate. Red here is the EPA drinking water limit, and you see throughout California, throughout the Salinas Valley, all kinds of drinking water wells are out of compliance with nitrate. This is a national problem. The USGS will tell you it is the biggest groundwater pollution problem in the US. USGS is installing a real-time surface water quality network. They are buying the sensors that we developed. It's a commercial product. They're expensive. They're over $20,000 a pop. And some states, Iowa has a much dense, they run in the middle of the summer, there's not a river or creek in Iowa that is not out of compliance with nitrate drinking water limits. So Iowa has its own very dense network. They'd be even denser if the darn sensors weren't so expensive. And, you know, so no one wants to put $20,000 sensors in the water at some point. So this is the opportunity. You know, even a drinking water nitrate sensor on the tap at home that glows green when you're below the EPA limit. And you can. So nitrate, huge interest in pH in the ocean. Atmospheric CO2 is going up. This is the Mauna Loa CO2 record. Same time CO2 goes up in the atmosphere, it's a gas, it diffuses into the ocean. This is the ocean record at Hawaii near Mauna Loa. It's going up in parallel with the atmosphere. When CO2 enters the ocean, CO2 and water react and it releases a proton. The ocean is getting more acidic. So here's pH. The pH of the ocean is going down. And that has huge environmental consequences. This is a very small decrease, but it turns out the ocean is not used to pH changes much at all, and some impacts. We'd like to measure pH. Only a few places we do it. This happens to be one near Hawaii. We tend to measure it near Bermuda. You measure it where there is an island big enough to have a university on it. And those are about the places where we have a record. So we would like to have our robots have pH on it. So we think of glass electrodes as a way to measure pH, but we have looked around and found a technology based on ion-sensitive field effect transistors built in the silicon foundry. We don't have a foundry at MBARI. We have worked with Honeywell to develop a pH sensor for the ocean. Honeywell developed it for industrial process control. We again, by two companies, papers in analytical chemistry on the system. That's an ISFET chip right there. And again, lots of papers on it. And again, you can measure pH in the ocean. This is the same record I showed down in the Weddell Sea. This is the high oxygen. When you have the plants grow more, here's the nitrate record. Plants took up more nitrate, and here is the pH record. When the plants grew, they took up CO2 by photosynthesis. If you take CO2 out of the water, pH actually gets higher. And this is the record of pH in under the sea ice. Again, where there's been ice. We can compute from pH the CO2 partial pressure in the ocean and compute in-gassing and out-gassing of CO2 to the atmosphere. And this is paper. Allison Gray is now an assistant professor at the University of Washington, took the profiling float pH data all around the Southern Ocean. Compute from pH, you compute CO2, and from CO2 and the wind speed, you can calculate the flux of carbon dioxide into or out of the atmosphere. We normally think of the Southern Ocean as a sink, that the CO2 flux is negative, that CO2 is going from the atmosphere into the ocean. That has been the story for decades of oceanography. The interesting thing is that no one gets on a ship and goes to the Southern Ocean in the winter, and especially you don't go down in this brown zone around the sea ice edge, because that is just not a good place to be in winter storms with lots of ice around. And so there really was not much data down here. There was really no data down here. And it was just sort of interpolated from summer to summer. And so Allison took the float data. These brown, these are different zones, the subtropics and then the subantarctic zone in red, and in blue is the polar frontal zone, and then ASC is the Antarctic sea ice zone and it is right on the edge of the ice. And historically, ships do transit up in the subtropics. And so this is what the ship flux would have been. This is what the floats would say. And within the error bars, a similar number. You get over here and totally different number than what we thought we were getting from ships. This is a flux, not from the atmosphere into the ocean. This is a flux contributing about 30% of the total flow of CO2 between the atmosphere and the ocean if you integrate it over the whole ocean. And it's just kind of turned out to be a revolutionary, eye-opening thing that we never saw because no one wanted to go there in the winter on a ship, but you can go in the summer and leave the robots and get a totally different story about what is happening with the atmosphere. So this is biogeochemical Argo. I've talked about chemistry, but huge interest in biology. So we measure chlorophyll. This is all commercial sensor-based work. Well understood. You can excite chlorophyll fluorescence. So you excite chlorophyll in plankton. This is not chlorophyll dissolved in water, but it's in the cells. You excite it at about 470 nanometers and measure the emission of light at about 700 nanometers. That's chlorophyll. We can do that. We can also measure the downwelling radiance of sunlight. If the float comes up in the daytime, the irradiance, so this is a radiometer with 4 different wavelengths, 380, 412, 490, and then a broad PAR, photosynthetic available radiation. And you measure the profile of light, and the more chlorophyll there is, the more plants there are, the shallower the light would penetrate. So this particular slope, if there were a lot of plankton, it might be a much steeper slope that looks like this. The 2 sensors should give you the same answer. They very often don't. That tells you a whole lot about the physiology and the biology of the plankton in the system. And our lab hasn't done a lot of work with this, but around the world, people are making, hey, again, profiling floats, seasonal modulation of phytoplankton biomass in the Southern Ocean. And partly I make the point that all of this data is publicly available. These are papers being written by people that, not in my lab, that are deploying the floats, but the data is available to anyone to write the papers. Papers in Nature Communications, in Science, in other Nature Communication on the bio-optical data. So anyway, to kind of bring this, so we have sensors, we have floats, we have sensors, we have experience, 18 years now of experience. This is the total number of BGC floats that have been deployed, the total worldwide. US is this trend down here, and the rest of the world, which is a bit more than half of the array, is the trend here. So, you know, right now there are about a bit under 400, 10% of the Argo array has chemical and biological sensors on it. And about half come from the US, and here they are deploying a float. So a robot that will run, you know, for somewhere between 4 and 7 years. So there's been, as this has progressed, there's been 10 years of community planning for a global array. I've made the point, I hope, that there are phenomenal things to learn about the ocean. There are imperatives to observe the ocean because changes are happening and we just don't look. And so starting in 2007, we wrote a paper. In 2007, all we had were oxygen sensors to put on floats. Since then, we've got the nitrate, we've got pH, we have bio-optical sensors. And as the array has gotten richer, we met a group, international group, met in Villefranche in 2016. There I am, and wrote an implementation plan for a global array. So you can find a copy here, biogeochemical-argo.org. This is an international plan. There were a number of studies done, OSSIES, which is an observing system simulation experiment. So you take a computer model of the ocean and subsample, which you presume to be the exact representation of what's going on in the ocean, subsample it to see how many floats, how many dots in the computer model you might need to reproduce the output. That's an OSSI. A variety of other things, and we said we need about 1,000 of these floats in the ocean with chemical and biological sensors. Historically, the US has done half. And if you did this, you would understand really transformative things about carbon cycling, OMZs, oxygen minimum zones. The ocean is losing oxygen. There are large areas of the ocean that essentially have no oxygen in them. And we would understand, are these zones expanding or contracting? How do they change? We don't know. We'd understand about nitrate, the nutrient flux, this acidification into the deep sea by sinking plankton. We'd understand a lot about phytoplankton growth and how all that tied into living marine resources, fisheries. Ocean takes up a lot of carbon, and as we've signed back into the Paris Climate Accord, which commits nations to targets for showing it to this, we can actually start to do global models that tell us whether nations are emitting or reducing their consumption. And as I said, the data is all available freely in real time. And so 2 years ago, mid-scale research infrastructure program came to be known in our group as MBARI. The MBARI program was announced, and it was for facilities, not research, but for facilities with a price between $20 and $70 million. We wrote a proposal to it for a global array of 500 floats, $52.9 million for that array. It's about $100,000 lifetime cost to run a float. And we wrote the proposal, 2 years of heavy lifting, and we were awarded in October of this year, the 52.9. And so we have just started the partner institutions, MBARI. We are the lead institution. I'm the director for the program. The University of Washington, Scripps, Woods Hole Oceanographic Institution, and Princeton are the other partners. This is really, a collection of some of the finest oceanographers in the world. Steve Reiser has been one of the founders of the Argo Array. Lynn Talley is, I would argue, the leading observational oceanographer in the US. Susan Wafels at Woods Hole has been a cornerstone of the Argo program since its inception. And Jorge Sarmiento is the most famous biogeochemical oceanographer in the world. NSF limits you to 5 PIs. These are the PIs for the proposal. There are a number of senior personnel that really did the heavy lifting: Heidi Cullen, Yui Takashita, George Matsumoto, Allison Gray, Sarah Perkey, Rue Nicholson, Andrea Fassbender, Todd Martz, and Roberta Hatinsky. All should have been PIs, but we're not allowed to be PIs. And so as we go forward, we're going to put 500 of these floats in the ocean. We use 3 different models, 3 different sources. Always, this weird manufacturing possibility that you get bad chips, bad batteries, bad, capacitors, whatever, and you're going to put something in the ocean for 7 years and they start failing. It's just poor form if that happens. So we spread the risk out by having 3 different sources of floats. It makes life more complicated, but also reduces risk. So Navis floats, Apex floats, and Solo 2 floats. And we start deploying floats go BGC this March. So this is our first deployment cruise. Because of COVID, it is extremely difficult to get on. We can't fly people around the world to load ships, so ships have to leave US ports. So this cruise leaves and doesn't come back to the US, it comes back, doesn't touch shore. We'll deploy floats down the Western Atlantic, comes back, deploys more floats, comes back to Woods Hole. So we can load in and out of Woods Hole, Massachusetts. Just as a point, in SOCOM, we're right now, there are a group of our scientists on a ship. They've deployed this line of floats. They're in a straight line because the ship went down this line. Last one was deployed yesterday. these guys are going to be on the ship for 80 days. They had to get on in the US and at 10 miles an hour. This is New Zealand right here. Just ridiculous. I mean, he's heroic to do this. But also awkward in the era of COVID. So challenges, scaling up BGC floats. You know, the project per NSF mandate is a facility. It is not a science project. It's like building a telescope. Now there need to be a community of users that come and use it. So we have to supply high quality data and really to detect the climate signals that we're all interested in. This is a 5-year proposal. We have to be good enough to be able to write a renewal. And so with that, I'm done. Glad to answer questions. That's kind of what a float looks like in the water. Thank you.
Thank you very much, Ken, for that talk. At this time, we'll move into questions. If you have a question, please, as Rebecca said at the beginning, react with either a raised hand or the yes check mark, and I'll call on you. Otherwise, feel free to message me a private message or put it straight in the chat and I'll read it out. Okay, first question is from Carla.
Hi, Ken. Great talk. I was just wondering what type of power needs these sensors have and how do you get around the challenges for power?
Yeah, a huge issue. So I said the floats will do about 280 vertical profiles, and the total power demand on a profile is about 15 kilojoules. So you can do 200 and something times 15. These floats have, so they're full of lithium, primary lithium batteries, which becomes a huge headache to ship them around the world, right? You've got a pressure housing full of lithium batteries that, you know, things go wrong. That's, you know, I mean, you've got like 5 or 6 megajoules in there that get released in a sealed housing. Bad things happen. So, so safety, huge concern. We tend to ship these only by ship. And people always ask me, do I want bigger batteries? My answer is no, I want more.
That makes sense. Thank you. And the lithium batteries, what do you, so when the float no longer works, these,
yeah,
what happens?
Yeah, great question, and I should have put a slide on the end of it. So the floats, it's easy for a float to go down, right? So we retract a piston, pull the oil. So gosh, I don't remember if I didn't show you how the floats actually work, but there's like a big syringe inside the float, and there's a little bladder outside. I push oil out into this, like inflating the balloon on a helium balloon. If I put a little oil into a flexible bladder, I've changed the volume of the float, but not the mass, and that changes the density. To go 2 kilometers, only 200, the ocean density is so constant that it only takes 200 milliliters of oil to inflate in the bladder to go up and down 2 kilometers, okay? So I can pull the oil into the float. That doesn't take much power. The float will go down to 2 kilometers. Takes a lot of power then to push that 200 milliliters of oil back out into the bladder to come back up. So when the batteries are weak and it's deep and it's cold, the floats disappear at 2 kilometers depth. The interesting thing, so, you know, we are, full disclosure, contributing to trash in the ocean at 2 kilometers depth. But if I do the math, so a float weighs 30 kilograms. A research vessel will burn about 10 tons of fuel a day. The trade-off is between, that's per day, 10 tons of fuel per day. The trade-off is between my 30 kilogram float that's going to be in the ocean for 7 years, making good measurements. If I ran a ship for that kind of time frame, I mean, the environmental cost of running the ship would be huge. So there clearly is a small cost of putting a float in the ocean that's 30 kilogram, 30 liter, 30 kilogram device. But if, you know, if you think you need that information to understand what's happening in the ocean, I happen to believe that. it's a relatively small price, and hopefully the information that comes back from these floats will lead society to modify its behavior enough that there will be a net gain in environmental, behavior that offsets the 30 kilograms from each one of these floats.
Thank you.
Our next question is from Leandra, who asks, thanks for the great talk. I'm interested if you run into the issue of having hard to measure regions in the ocean with low current that are hard to get a float into.
Yeah, so as I said, floats aren't anchored, they drift. In the Southern Ocean, we have floats that will go almost all the way around the world. There are strong currents, and no land mass. You can go all the way around the world. There are places, I mentioned that there are large regions of the ocean where oxygen is disappearing because the currents are very weak. They don't get reoxygenated there very, very often. And so if I put a float in one of those regions, it'll stay there. So to get a float into one of those regions, I have to go there. The hardest one of these places is the Arabian Sea, just because for years we couldn't get a ship in there because of piracy. There was no US research vessel that would get anywhere near the Arabian Sea. Just wouldn't happen. We were able to get some ships in from US Navy destroyers. But those regions are hard to sample. Otherwise, the biggest problem in general for building a global array is bumming a ride. Literally. I mean, we don't have ship time. We are bumming rides from the rest of the world. If we had to pay for the ship time, my 50 million would be probably 200 million, because these $30,000 to $50,000 a day ships, would just kill our budget. So, we're hitching.
Our next question is from Professor Bayer, who says, I have read that iron is also an ocean fertilizer. Is this a parameter that needs to be measured?
Yeah, I was kind of expecting somebody to ask me what are the things we need to measure. And uniformly, if somebody does ask, they want to measure iron. So, for decades, I made my living as an iron geochemist, measuring iron around the world. I did some of the first iron fertilization experiments where, in fact, we did show that iron is an essential fertilizer. You can go out and spread iron on the ocean and just make phenomenal plankton blooms. So with that as a caveat, I have no idea how to make an iron sensor. I want one desperately. I just do not know how to do it. And it's complicated because, iron is in a myriad of forms in seawater, super insoluble. The dissolved concentrations are super low, less than 1 nanomole per liter. Most of that is strongly complexed by siderophores, bacterial ligands that bind iron. And there are plankton, some that just get the siderophores, some that just get the dissolved iron, some that get the particulate iron. There's dust and so on falling in. And you really want to measure all of those if you want to do a good job. And concentrations are low, the analytics is hard. And so Tom, go for it. But
Okay, we have another question. This one from Pingyu, who says, thank you for the great talk. I'm curious if you plan to include ways to detect and measure microplastic particles in the future.
There are, haven't gotten to that point yet, but there are optical microplastic sensors people are working on. It wouldn't be a ridiculous thing to be able to do it. It's not at the top of our list. There are some other actual fertilizers. So Tom mentioned other fertilizers. So I can measure nitrate. So in my world, nitrate's the most important fertilizer, right? Although there are other people that will say iron is the most, and there are other people who will say dissolved silica. So diatoms, right? You've heard of diatomaceous earth. Diatoms are these little phytoplankton, and they're limited by silica. So they make a silicious test, a shell, and not enough silica. And especially there's also a saying in oceanography: diatoms equal fish. They're big phytoplankton. They are the food chain that really leads to fish production. And so I'd like a silica sensor. I'd like an iron sensor, and and so microplastics would be on the list, but they're not going to be number one. On the other hand, maybe it's easier to do microplastics than iron. So maybe we should think about that, because there's imperative, there's also, ability, and somewhere in there you have to balance those two things.
I have a question on the lifetime of the floats. What limits the overall lifetime of the float? If it's a power issue, is there a way to recharge it?
Well, it is power. So, I've got my 5 megajoules of lithium primary batteries, 15 kilojoules per profile. You do the math, and the batteries go dead, it dies. There is a group at the company, C-Trek, that have built a thermal energy extract in the ocean, and they have demonstrated that you can take a float and make it essentially make a perpetual motion machine. They have a material that expands as it warms up. It expands and pushes oil and just essentially drives the generator and recharges rechargeable batteries, right? And problem is that I put these on a float. I've just tripled the floats are expensive. So 60, 70,000 bucks a pop for a float. And now I make it $150,000 device. And is it better for me to have one device that might run forever, but I'm not, at some point the sensors will fail, they'll foul. And is it better for me to have one device that runs sort of forever, or 3 devices to spread the signal out for the same dollar amount? There is, you can get out your Excel spreadsheet and do something about what's better. But right now we have been conservative, and we have not gone to these energy extraction devices. Although they're super cool, they have demonstrated them, they work. They just adds, another element to the cost that right now maybe it's just better to go simpler.
Thanks. Our next question is from Adele.
Hi, Ken. Thanks for the talk. My first question is that it seems like a really good opportunity with all of these Argo floats in the water to maybe get an idea of like what animals are in the water. So like, could you have some sort of acoustic sensor that can listen for whales or other animals?
Yeah, there's a variety of things that are happening. We actually do put acoustic sensors in some of our floats. We do that not so much for animals, but we when the floats park for 10 days at 1 kilometer, you can hear the waves break, and it's a really good acoustical wind speed sensor. So they're out in the middle of the ocean. No one's measuring wind speed. They want it for meteorological models. They kind of sense it from satellites, but we actually do a better job by listening to the crashing of waves. You also get rainfall. You can also listen to the pitter patter of rain at a kilometer depth. So totally doable. We keep talking about expanding this to listening for, essentially putting a fish finder on a float is the way we describe it. It's certainly doable. European colleagues are really pushing a technology called the underwater vision profiler. So although fish are interesting and so on, it's, you start with the tiny phytoplankton up around 1 millimeter in diameter. You want to know about the zooplankton and so on, the next step in the food web, and the underwater vision profile just provides you a phenomenal record of that. So they're, starting to put these on floats right now. The data streams, so iridium is great, but it's still only about the real turnaround in iridium is about not better than 1 kilobaud, 500 baud. To get a lot of imagery back is just not feasible over low Earth orbit, low power telemetry systems. So they have to recover the floats to really get the data, and that is not how you build a global network. You build a global network, you just put the floats in one way, you never get them back. They're great for what we call, you know, sort of experimental projects. You put it in, you come back a month later, you get it, kind of thing. And so needs to be some work done, needs the data bandwidth is an issue, but there's huge interest, you know, in either acoustical sensors. So when we put these acoustical sensors for wind speed, all the processing happens afloat. We don't send all that data, we just process it down and send a wind speed number back.
Okay. Cool. Then my other question was, you talked about how your floats calibrate for the oxygen levels, but what about the other ones? Like, how do you know they're, you know, giving you the correct values?
Yeah. So huge problem. A number of papers we've written on this. So the oxygen sensor we can calibrate in the atmosphere, and we know, you know, so we get a really good oxygen number. The nitrate or pH sensors, so frankly, most of our interest is the upper ocean where the biology is happening, but we profile to 2 kilometers if for no other reason to recalibrate the sensor in deep water. So we've put a lot of effort into showing that when there is an error in the sensor, it's an absolute offset, you know, plus or minus some constant number, and that constant would apply to a whole profile. So there is ongoing in the ocean programs that resample the ocean depths at about a decadal interval called GOSHIP, Global Ocean Ship-Based Hydrography Program. And the data at 2 kilometers is really quite stable. And if you have oxygen, you can predict what nitrate is at 2 kilometers almost as accurately as I can measure it. So when the float goes to 2 kilometers, I look at my sensor value, and then I take this ship-based data and the oxygen and predict what nitrate ought to be. And if they don't match, we actually start adjusting our data. If we see the sensors start to drift away from these deep reference values, we adjust. And so, you know, and do the same thing for pH. And Argo has always done the same thing for salinity. The salinity sensors in the float, again, none of these sensors are perfect. They all have some element of drift. The oxygen sensors, although really great, still have an element of drift. And but we can recalibrate them in the atmosphere and then use them to predict in the deep ocean, you know, what our other sensor values ought to be based on this history of ship-based measurements, and adjust.
Okay, great. Thank you.
I think we have time for 1 or 2 more questions. So the next one is from Pratap.
Hello, Professor. Thank you for the talk. I like a lot to know, and glad that there is a sensor network available to everyone, the data. I have a question, or I would say two. Like the first is, since the sensors are so deep, but do the data give us a prediction or a parallel of, okay, this area there is a greater degradation of the coral reefs, and like around this area there is less pollution, less degradation, or the degradation of coral reef is more of a global thing, it cannot be localized?
So huge issue with coral reefs. So you know, we tend to not to get into the coastal zone. These floats, you know, want to be out in essentially water deeper than 2 kilometers, and when they get in close, they hit the bottom and tend not to get too in. They tend not to get shallower than 2 kilometers. So we don't do coral reefs. But the whole point of this eventually is to assess, to answer your question, which areas of the ocean are doing good. And if you look at the computer models of future change, some areas will benefit. At very high latitudes, there aren't so many plankton because the winter storms, mix the plankton down. If the winds drop in a warmer climate, there'll be more plankton, there'll be more fish, there'll be, you know, might be healthier. Flip side, in the water near Hawaii, I showed those all these little nitrate uplifts. If the water gets warmer, it takes more energy, it's less dense, so it'll take more energy to lift nitrate up. The ocean might get poorer in those areas. We would like to be able to answer that. Right now, we're just starting this. So the way I describe this, we are taking the vital signs of the ocean. We are measuring, it's like taking your temperature, your pulse, your blood pressure, your heart rate. We're trying to figure out, right now, we don't even know what those vital signs are. Is the pulse rate of, blood pressure of 120 over 80 good or not? We don't know. We don't know. So right now we're establishing the baseline. And if we do this long enough, we will see change and we will be able to answer your question. But right now, I don't even know if the ocean is happy or not. I'm just trying to find out what it is, if that makes any sense.
Totally.
Yeah.
And another question is like, is there a security issue with these gliders or these bots? Like, can somebody steal it? Or I mean, if somebody wants to steal them or not? Like,
so the floats come up for only 15 minutes when they use iridium. And by the time, if one came up right next to the ship, by the time you got organized to get a small boat in the water to grab it, whatever, it would be gone again, right? But before we had iridium, the floats used a system called Argo. So this is the Argo array, but there was a satellite communication system called Argos. And Argos, so I complain, that iridium's like, 1 kilobaud, right? Well, Argos was 10 baud, 10 bits a second, okay? And the floats, not only that, but it was one way. And so you never knew if the satellite was getting your signal or not. So you had to stay at the surface and send your data over and over and over again, just to be sure that it got out. And in that case, the float would be on the surface for 12 hours sending, and people did get them. And they would end up in, so you do get a GPS signal, right? And so you'd find floats, and all of a sudden the float would take off at 10 miles or 15 miles an hour, or whatever that way, and it'd go to a port, and then it'd be 60 miles an hour going down the road, and right where it was. So so there's an obligation, right? These things are full of batteries, and onshore in people's hands, bad things can happen. They found one of these in a pile of trash that was about to get burned, which would have, been a mess. And so we have an obligation when they hit the shore. This is part of the international agreement, is to get them back. Interested as a whole, there are people, they should do like a reality TV show on getting floats back. But in Somalia, right? Floats have gone ashore in Somalia. So what do? There are missionary groups. I mean, I would never go to Somalia, but there are missionary groups in Somalia, and you can hire them. They'll go get your float. I mean, I don't know, I hate to say what they're smoking, but but they'll do it for you. So you pay them and they'll go get the float for you. Kind of, reality TV. Crazy.
Yes. Nice. Yeah, that's what I was like, some anecdotes of getting back. Nice.
Yeah.
Thank you.
There are still some floats in places where nobody will go, but but in principle, we are obligated if they hit the beach to go get them.
Makes sense. Nice. Great talk, Professor. Thank you.
Great. So with that, let's please give Ken another round of applause. Thank you, Ken, so much for your time and for your talk. And thank you, everyone, for attending. And with that, we will conclude the seminar.
Okay. Thank you very much. It was a lot of fun. I'm glad to answer email or whatever. So, all right.